we have discussed about this secretions from the salivary glands, then secretions from the stomach, okay, and next is the secretion from the pancreas, okay, and then we'll discuss about liver. What are the secretions from the liver and why these secretions are required? Okay, then yeah, so as you all know, where is pancreas located? It is located just parallel to the stomach, okay, beneath the stomach, and it is a large compound gland that you know. Compound gland means it is consisting of numbers of endocrine as well as exocrine glands, okay. So here we are going to discuss about the exocrine part, okay. The exocrine part, again, it is a compound exocrine gland because the numbers of branches are there in different portions. The glands are connected by the branches of the ducts, okay. So because of that, they are called a compound gland. And if we look into the internal structure, the structure of the cell, this pancreas resembles to that of the salivary glands, okay. And here one term is a pancreon. You might have not heard this term. A pancreon means like a salivon. Salivon is a functional unit of salivary gland. Just like that, a pancreon is a functional unit of pancreas. That means the whole of the function of the pancreas is going to be done by each of these pancreons, okay. So the pancreon is going to have its secretory functions. The secretions, say, in the form of the enzymes, or the secretions in the form of the salts, okay, juice part. So a pancreon is a functional unit of pancreas, and it consists of pancreatic acini and the ducts, okay. So this pancreatic acini, if you look into the anatomy of the pancreatic acini, so this figure is showing, it is just depicting, okay. So in this figure you are seeing only one acinas, another acinas, another acinas, and their secretions are poured into this duct, okay. So it is not only like this, but there are multiple number of acini, okay. Just to make you understand, here the picture is showing only one acinus, here one acinus, one acinus, like this, okay. So multiple number of acini are connected by the intercalated ducts. Then the intercalated ducts will be connected by interlobular ducts. Then interlobular ducts will be connected by the lobar ducts. Then the lobar ducts will fill into the will be connected to the pancreatic duct, okay, the main pancreatic duct, and those secretions will be poured into the intestine. So that is about the anatomy of the pancreas, okay. So here what we are concerned is who are the secretions from these pancreatic acini or from the ducts, and what are their roles. So that is our interest here, okay. So here so the pancreatic acini secrete the pancreatic digestive enzyme. Means these acini and the ducts are having their respective roles, okay. That means the acini will be secreting the enzymes and the ducts will be secreting the fluid part. That fluid part mainly consists of the sodium bicarbonate. Means they are in the form of the pancreatic juice that consists of numbers of salts, and of that one main component is sodium bicarbonate. Okay, so these the secretions of the pancreas means who are the secretions of the pancreas. One is the pancreatic enzyme and another is the pancreatic juice. That pancreatic juice is consisting of the numbers of salts, and main component of that pancreatic juice is the sodium bicarbonate. Okay, so after the secretion from the acini, the pancreatic enzymes will flow into the lumen of the acinus. Then that will be collected by the intercalated duct. Then in many intercalated ducts will join and form the interlobular ducts. Then many interlobular ducts will join and form the main pancreatic duct, and that pancreatic duct will empty their secretions into the intestine. Okay, so the opening of the pancreatic duct and the opening of the bile duct is the same here. Okay, so they will open together in the intestine. So at that opening, all the secretions will be poured over here. So what we have learned earlier, so the contents from the the partially digested food. Some amount of the starch digestion has started in the mouth, okay, in those animals which has salivary amylase. Then the food goes to the stomach, and in the stomach the digestion of the protein starts. Okay, so the contents in the stomach which is mixed with the acid of the stomach will now come out of the stomach and will be reaching into small intestine. So by the time the food contains the acid kind which has now reached to the intestine will make the environment in the intestine acidic. Okay, so but these foods are partially digested. They need to be digested further. For that, for the digestion of the food substances, for digestion of the further digestion of the starch, further digestion of the proteins and digestion of the fats, they require many other enzymes. Okay, actions of many other enzymes, and those many other enzymes will be secreted by the pancreas. So majority, almost all of the secretions, almost all of the enzymes coming from the intestine require an alkaline medium. You get the point? Do you get the point? Hello? Yes. So the actions of these enzymes to act on the carbohydrate, to act on the protein, or to act on the fat, okay, requires an alkaline medium. But the food which has just now come from the stomach is highly acidic. Okay, so to neutralize that, the bases, the bicarbonates, the salts, okay, are secreted from the pancreatic ducts, and they will be poured and mixed with the acid chyme, and these will change this medium in the intestine into an alkaline medium. Okay, so what for pancreatic secretion is required? One is the secretions from the pancreas, the pancreatic juice, to make the environment in the intestine alkaline, because many other enzymes which are present in the intestine itself, or the enzymes which are coming from the pancreas, needs an alkaline medium, alkaline environment. Okay, so in addition to that, numbers of the enzymes for digestion of all the nutrients, say for digestion of the starch, pancreatic amylase will be reaching here. For digestion of the protein, numbers of protein digesting enzymes, say trypsin, chymotrypsin, then numbers carboxypeptidase A, carboxypeptidase B, like this. Okay, so they will be coming here, say for digestion of the fat, then what pancreatic esterase, the pancreatic lipase. Okay, they all need to be secreted to digest the food which has now reached the intestine. Okay, so why there is secretion of the pancreas? One is to neutralize the acid so as to enable to the enzymes to act on the partially digested food for further digestion. Okay, another is to digest the proteins. Protein digesting enzymes will be brought here. For digesting the fat digesting enzymes will be brought here. And for digesting the carbohydrates, the carbohydrate digesting enzymes will be brought here. Okay, they will be secreted from the pancreas and be poured into the intestine. Is it okay up to this? Okay, then I have listed out, okay, I have listed out here the enzymes, okay, the enzymes and their activators and where they act on, okay, and what are the products formed in the tabular form here. So you just remember one by one. Say from the salivary glands, salivary amylase comes, and who is the activator of the salivary amylase? It is the chloride. Okay, and they act on starch. Okay, then alpha limit dextrins, then maltotriose and the maltose will be formed. They will act on the 1,4 linkages, 1,4 glucose linkages. Okay, then from the lingual glands, the triglycerides, okay, they with this lingual lipase will be secreted and they will act on to the triglycerides. Okay, then from the stomach, pepsin, then from and also the gastric lipase. Okay, so these are the list of the enzymes I have listed. So simply you have to remember here one main important enzyme coming from the exocrine pancreas is trypsin. Okay, so what you have to remember here, trypsin as such is not secreted from the exocrine pancreas, but it is secreted in the form of trypsinogen. The function is just like pepsin is secreted in the form of pepsinogen and becomes active and becomes pepsin. When pepsinogen is mixed with ICL or already mixed, or the pepsin which is already mixed with the ICL in the stomach, something like that, trypsin is secreted in the form of trypsinogen, and this will be activated only after it reaches the intestine where the enzyme enteropeptidase is present. Okay, so this you remember, okay? So this is an important point, that one of the most important enzyme present in the pancreatic secretion. Pancreatic enzyme is trypsin, but trypsin is not secreted in the trypsin itself, but it is secreted in trypsinogen. This trypsinogen is activated by the enzyme enteropeptidase. Enteropeptidase is present in the intestine. So only after the trypsinogen comes into the intestine, it will become trypsin. Why? If trypsin is present in the pancreas itself, then the trypsin may cause the destruction of the pancreas itself, because this is a very strong protein digesting enzyme. Okay, so that is inhibited. Okay, that is inhibited, that is prohibited, because trypsin is secreted in trypsin in the form of trypsinogen, and it becomes trypsin only in the exposure to enteropeptidase. Where enteropeptidases are present, they are present only in the intestine. Okay, so that trypsin will act onto the proteins and many other polypeptides. Okay, so by the time now, when trypsin is already present in the intestine, many other enzymes coming from the pancreas will also be activated. Say, trypsin will activate chymotrypsinogens to trypsin. Okay, procarboxypeptidase to carboxypeptidase A, procarboxypeptidase B to carboxypeptidase B. Okay, so trypsin becomes available only after trypsinogen comes in contact with enteropeptidase. Okay, you remember this point, and trypsin is one of the most important and the most prominent, okay, enzyme, protein digesting enzyme present in the pancreatic secretion. Okay, so many other enzymes are also present in the pancreatic secretion. That is elastase, which is secreted in the form of proelastase, becomes active in presence of trypsin. Okay, another is procolipase. Okay, that becomes colipase in presence of trypsin. Then the many other enzymes which are pancreatic lipase, bile salt acid lipase, cholesterol ester hydrolase. Okay, so these are the fat digesting enzymes. Okay, then the pancreatic amylase, same like the salivary amylase, the pancreatic amylase, it becomes active in presence of chloride, and they act on starch. So same like salivary amylase, they will be acting onto the alpha 1,4 linkages. So do you understand what is alpha 1,4 linkages or beta 1,4 or beta 1,6 linkages of glucose? What is that? What is alpha? And what is beta? And 1,4?
When it is glycosidic linkages.
Between, if it is 1,4, then the link is between
First and fourth carbon.
first okay, carbon of one glucose and with the fourth carbon of the another glucose, right? So like that, so they will be acting at those 1,4 glycosides. Okay, though they will break the 1,4 glycosidic bonds. Okay, so they will act onto them and they will form the alpha limit dextrins. Okay, then ribonucleases, then deoxyribonucleases, then phospholipase A2, which is this what secreted as prophospholipase A2. Okay. Okay, that means you see here, trypsin is activating all other enzymes, okay, protein digesting enzymes. So the presence of trypsin is very, very important. So when trypsin will be present only if it is exposed to enteropeptidase. So where the enteropeptidase is present, it is present in the intestinal wall. Okay, so on the walls of the intestine, they are present. So once trypsin is present, other substances containing the protein digesting enzymes, they will become activated. Okay, then who will regulate? Next point is who will regulate the secretions of intestine of pancreas. So who will regulate? If the foods are not present, if this food, the food which has to come from the stomach to the intestine, if they are not there, will there be secretion of the pancreas? Will it require pancreatic secretion? No, it is not required. But who will sense it? So the presence or the absence of the food items, the partially food items in the intestine will be sensed through the enteric nervous system. We have learned about the enteric nervous system, right? So in the enteric nervous system is something like autonomic nervous system, isn't it? So in the autonomic nervous system, there are sympathetic and parasympathetic nervous system. Again, when we talk of the nervous system related to digestive system, you think only about the parasympathetic nervous system. Sympathetic nervous system stimulation means inhibition of digestion. Stimulation of the parasympathetic nervous system means stimulation of digestion. Okay, so whenever there is presence of the food items in the intestine that has been expelled from the stomach, this will be sensed through the enteric nervous system. So it will go through a reflex to the brain, and through the autonomic nervous system, and it will stimulate the parasympathetic part of the nervous system of the autonomic nervous system. We can think of the autonomic nervous system or the enteric nervous system. Okay, so the enteric nervous system is going to sense it, and I told you that the enteric nervous system works in conjunction with the autonomic nervous system, both sympathetic and parasympathetic. So here, the branches of the vagus, okay, the branches of the vagus nerve, and that also the parasympathetic branches of the vagus nerve will release, they will be stimulated and they will release acetylcholine. So do you remember the postganglionic nerve terminal in the parasympathetic nervous system secretes which type of neurotransmitter? Which type of neurotransmitter is released by the postganglionic nerve terminal of the parasympathetic nervous system?
Ma'am, cholinergic.
Cholinergic. Yeah, postganglionic nerve terminal is cholinergic. Cholinergic means they will be secreting acetylcholine and acetylcholine-like substances whose functions are similar to that of the acetylcholine. Okay, so you think of acetylcholine here. Okay, so you just know that the parasympathetic nerve terminals will release will lead to the release of acetylcholine. So whenever there are food substances present over here, the enteric nervous system is going to sense it, and through the stimulation of the enteric nervous system, it will stimulate the parasympathetic nervous system, and the parasympathetic nervous system will release acetylcholine, and that will lead to the stimulation of the what? The acini, okay, the pancreatic acini for secretion of the enzymes. That means the sense that the food is present over here and they need to be digested will go to the pancreatic acini, and they will be stimulated, and they will lead to the secretion of the more of the enzymes. Do you agree? Is it possible or not? Yes, yes, it is possible. In another side, what happens when there is distension of the pyloric region? When there is distension of the pyloric region, because as the food is passing towards here and the sphincter has not yet opened, okay, the sphincter which opens the stomach contents to the intestine, if it has not yet opened, but by the time the food substances are present over here, it leads to the distension of the pyloric region. So the distension of the pyloric region, what leads to it leads to the secretion of the cholecystokinin. Do you remember that part? Yes, yes, I told you that distension of this pyloric region, it leads to the stimulation of the cholecystokinin. Okay, and when another point, by the time the sphincter is open, the food is present in the intestine. Now, this food is acidic in nature because the contents coming from the stomach is highly acidic. So by this time, the acid sensations will also give a signal for stimulation of secretin. Okay, because the secretin is going to stimulate the secretion of the pancreatic ducts. That means that acid present in the intestine, the food present in the intestine, the distension of the pyloric region will lead to the stimulation of secretin and cholecystokinin. More and more of the secretin and the cholecystokinin secretion from the duodenum. Then what will happen? That will come through the blood, and secretin and cholecystokinin will now act onto this pancreas. Here, secretin stimulates fluid and the bicarbonate secretion. The fluid and the bicarbonate secretion will be coming mainly from the ducts, particularly the smaller ducts. The smaller ducts will secrete the fluid and the bicarbonate, and the cholecystokinin will stimulate the acinar secretion. So I told you about the acini in the salivary glands. Just like that, these acini, the pancreatic acini, they will be secreting their secretions, mainly the enzymes. So these enzymes which are present in the acini, in the zymogen granules, they will be secreted from the epithelial cells of the acini and will be poured into the lumen, and then will be collected by the ducts. Okay, so these whole are regulating the secretion of the pancreas. So here what happens, the acid from the stomach releases the secretin from the duodenal walls, and fats and the amino acids from the fats and the amino acids, where they will be present in that food only. Okay, the food which has come from the stomach, they will stimulate the release of more of the cholecystokinin. So now the cholecystokinin will stimulate enzyme secretion, enzymes for the carbohydrate digestion, enzymes for protein digestion, enzymes for fat digestion. They will be poured now through the duct into the intestine. Okay, so that is how. So there are three mechanisms involved for regulation of the pancreatic secretion. One is the secretion of acetylcholine from the parasympathetic nerve terminal of the vagus nerve. Another is secretion of cholecystokinin. Another is secretion of secretin. Is it okay? Is it okay? There are three factors, okay, who are regulating their secretion. So here, someone may ask the differences of secretin and cholecystokinin. In the previous class, when we discussed about the acid secretion also, I told you role of secretin in acid secretion and role of cholecystokinin in acid secretion. Okay, that row, and here these rows, including all these rows, you can make a table citing the differentiating points between secretin and cholecystokinin. You can make your own. Okay, so someone can easily ask you that someone may ask simple questions, say like how the pancreatic secretion is regulated, then it is okay. But someone asks you what are the differences between secretin and cholecystokinin, then you have to think. Okay, so for that you make a table differentiating the secretin and cholecystokinin. Okay, so here one more point, important point is that these pancreatic secretions, particularly the protein digesting enzyme trypsin, okay, so trypsin secretion is usually kept inhibited by trypsin inhibitor. Okay, so trypsin inhibitor is a peptide, okay, so which is present in the intestine, which inhibits the trypsin. Okay, so once trypsin inhibitor is inhibited, then there will be secretion of the what I'm telling. So once this trypsinogen comes with the enteropeptidase, then only it will be converted to trypsin. Otherwise, trypsin is kept inhibited by the trypsin inhibitor. Okay, so this point you should remember. Okay, and one more important point is that this is the trypsin which is stimulating other protein digesting enzymes. So this you have to remember few of these points, and differences between cholecystokinin and secretin you make yourself. Okay, then comes the secretion of bile by the liver. So you all know bile is secreted by the hepatocytes. Hepatocytes means these are the cells of the liver. Okay, so the cells of the liver are secreting the bile, and this bile will be collected through the ducts and they are stored in the gallbladder. Okay, so as and when there will be requirement of more of the bile, stimulus will go to the liver for more secretion of bile, or the stimulus will go to the gallbladder for release of the bile through the bile duct. Okay, so bile consists of bile acids, which are steroid acids, means these are made up from steroid. Okay, then the bile salts, which are conjugated to glycine and taurine. Okay, so bile consists of bile acids and the bile salts, and the who are those bile salts? The names of the bile salts, they are the taurocolic acid and the glucocolic acid. Okay, so or we can say sodium taurocholate or the sodium glycocolate, like that. Okay, so these are the names. And then what are the functions of the bile? Okay, so bile plays important role in fat digestion and absorption. How? The bile is going to break the fat molecules, the large fat particles, into smaller fat particles. Okay, those smaller the making of the large fat particles into small fat particles is called the process of emulsification. So the process of emulsification is only possible in the presence of bile. So it helps in the digestion of fat, because these large fat particles cannot be acted upon by the fat digesting enzymes. They cannot be absorbed through the intestinal walls. So before they are digested by the fat digesting enzymes, they need to be broken into smaller fat particles. That process is called emulsification. Okay, so after the emulsification, there will be easy absorption of the digestive fats. Okay, then the bile also serves as a means for excretion of bilirubin and excess of cholesterol also. So you remember how the RBCs were lysed, and after that the bilirubin was formed, and how the bilirubin is excreted from the body. So this is also formation of bile is also a means for excretion of bilirubin. Okay, so who are and how bile secretion is regulated? So this is another important point. So in the liver, okay, in the liver, there will be the accumulation of the bile acids, okay, which will stimulate, okay, which will stimulate the secretion of the liver. The parenchyma of the liver is made up of the hepatocytes, okay, and which are connected by the ducts, okay. So smaller canals. So the bile acids which are coming through the blood circulation will stimulate the liver parenchyma for secretion of bile by the hepatocytes. Okay, so now this synthesized and the secreted bile will be coming through the hepatic duct and will be stored in the gallbladder. Okay, so the bile is stored in the gallbladder until and unless there is a requirement of bile in the intestine. Bile will remain here in the gallbladder. So removal of bile from the gallbladder will only be possible through the opening or through the relaxation of the opening where the bile duct opens into the intestine. Okay, that opening is called the sphincter of Oddi. Okay, so here what happens when the food, okay, is mixed with acid and it is now coming out of the stomach and reaching to the intestine, there will be stimulation of cholecystokinin. There will be stimulation of secretin, right? That I told you just now, okay? So that cholecystokinin or that secretin is going to help in the secretion of bile and also in the release of bile. First of all, the cholecystokinin, which secretion is stimulated by the presence of the what? By presence of the proteins or by presence of the fats. Okay, that stimulates that cholecystokinin secretion, which increases due to the presence of the protein here, presence of the fats here. Okay, this will come through the blood circulation. This is cholecystokinin is a hormone, so this will come through the blood circulation and will lead the gallbladder to contract. Okay, while gallbladder is contracting, there will be relaxation of sphincter of Oddi. Okay, so these two actions are brought by cholecystokinin. That means by the time When the food substances are present over here, there will be secretion of cholecystokinin, and that cholecystokinin will lead to the contraction of gallbladder. When the gallbladder is contracting, then there will be opening of the sphincter where the bile is released into the intestine. Okay, one action over here. And also the gallbladder will contract when there is vagal stimulation. Vagus means the parasympathetic branch of the vagus. Okay, so then when there is vagal stimulation, where from this vagal stimulation will come? When the foods present in the intestine leads to the distension of the intestine. When the foods present in the pyloric region leads to the distension of the pyloric region. So all these are going to stimulate the vagus, and when the vagus is stimulated, it also leads to the contraction of the gallbladder. So the function of cholecystokinin contracting the gallbladder, it is aided by the stimulation of the vagus also. So the contraction of the gallbladder, it will lead to the emptying of this gallbladder and releasing the bile. Okay, and that bile which is coming through the duct will be poured through the opening of the sphincter of Oda and will mix with the intestinal contents. So here the bile will reach over here. Side by side, the foods which are present over here, it also stimulated the secretion of secretin. Okay, so that secretin, what it will do, that is the secretin will come through the blood circulation, and that will stimulate the liver ductal secretion. Okay, the hepatocytes will be secreting the bile, and other fluid part, okay, will be stimulated by the ducts. Okay, so they are all connected by the ducts, so they will be secreting the fluid part. Okay, so the hepatocytes will secrete the bile and the ducts will secrete the fluid part. Okay. So now the liver will have more and more secretion of the bile when secretin is more, and when the cholecystokinin is more, there will be more contraction of the gallbladder and more release of bile into the intestine. So now that bile which has now reached to the intestine will be helping in the digestion of the fats. Okay, is it okay? Okay, so for today I told you about the pancreatic secretion and the liver secretions from the hepatic secretion. Okay, secretion from the liver and how they are regulated. So they are all interlinked. Three main points are there. One is distension of the intestine leading to secretin and cholecystokinin secretion. Okay, presence of the acid into the intestine leading to cholecystokinin and secretin. Okay, that means acid leading to secretion of the secretin, distension leading to secretion of the cholecystokinin. Okay, acids as well as the distension leading to the stimulation of the vagus. So these three are the main important points which are regulating the secretion of the acid here. Okay, and in addition, many other factors are also involved for acid secretion, but for the secretion of the pancreas but for the secretion of the liver, these three are the main regulating factors. Okay, so you have to think when you read these three portions. Okay, you try to link them. When you discuss about the acid, when you discuss about the pepsin, when you discuss about secretion of pepsin, when you read about the secretion of liver, when you read about the secretion of pancreas, these three parts, okay, you try to link them. So you will know better and it will be easy to explain. Okay, is it okay? Anything you want, any explanation on it? Any clarification? So the explanations are given in your notes. I have given here in the form of the diagram for easy understanding. You can go through those notes. If you feel the need for more explanation, then we can discuss again. Okay, in the next class. You can ask now also if you have anything. And one more point is you note down 5-6 differentiating points between secretin and cholecystokinin. That is your homework. From all these discussions, you note down the differentiating points between secretin and cholecystokinin. You want to ask anything? If not, then let's end over here. You can leave the class.
Thank you, ma'am.
Thank you,
Thank you,
ma'am.
ma'am.
Okay, you read, okay? You have to read. You read the stomach secretion, liver secretion, and pancreas and try to link them one another. If you read only one, it will be very confusing. Okay?
Thank you, ma'am.
Okay.